Hello everyone, welcome to today's webinar. I'm Melissa O'Mara, Forensic Science Consultant in CNEN Media Group. I will be moderating today's event. This webinar is titled Protein Higher Order Structure Analysis, Essential for Achieving Quality by Design in Biotherapeutics, and it is sponsored by RedShiftBio. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of your slides on the screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars. Each webinar will be archived at CNEN online after the live webcast. The presentation today is sponsored by RedShiftBio. RedShiftBio is a provider of analytical instrumentation for the R&D and manufacturing of protein therapeutic drugs and has developed Microfluidic Modulation Spectroscopy, MMS, to probe protein biophysical structure. This analytical technique measures stability, similarity, concentration, secondary structure, and aggregation in a single automated analysis, replacing the requirement to run samples on multiple instruments. During the presentation, we will be hearing from Ms. Susana de Jesus Acosta and Dr. Patrick King. Susana is a senior researcher in the biotechnology department at Biokit R&D in Spain. She earned her BS in chemical engineering from the Universidad Autónoma de Santo Domingo in the Dominican Republic and her MS in plant biotechnology from Ohio State University. Having returned to the Dominican Republic to work as a researcher studying bioactive plant molecules, Susana then moved to the Netherlands and spent nearly the next decade working for Synthon on the synthesis and biocharacterization of antibodies and antibody-drug conjugates before taking up her current position. Susana's extensive experience with a very broad range of spectroscopic and other biophysical techniques towards the elucidation of protein structure makes her ideally placed to highlight the utility of MMS in the Protein Biotherapeutic Toolkit and give this webinar presentation alongside RedShiftBio to describe work on a collaborative project using MMS to identify good and bad candidates for manufacturing latex products, undetectable using current technologies. Dr. Patrick King completed his PhD at the University of Bristol in 2011 on the design and biophysical characterization of self-assembling peptides. He spent 5 years as a postdoctoral researcher at the University of Manchester in the areas of peptide and oligonucleotide chemistry, the design of novel biosensors, and the design and characterization of self-assembled peptide and DNA composite nanomaterials. Subsequently, he worked as a bioscience field application specialist at Malvern Analytical for 2 years, specializing in a number of biophysical techniques before taking up his current position as European Senior Field Application Specialist at RedShift Bioanalytics in 2019. So, with that being done, let's get started. I will now turn it over to Patrick.
Thank you so much, Melissa, for that kind introduction. Hi everyone, my name's Patrick and I'm a field application specialist for RedShift Bioanalytics. As has been mentioned, I'm going to give you a talk today on Microfluidic Modulation Spectroscopy, or MMS, which is a revolutionary new type of infrared analysis technique. And this has been brought to you on the AQS3 Pro platform, which is a system that we launched last year. I'm going to start the talk just by giving you a bit of background into the company itself. I'm then going to go on to describe how MMS can be applied and fits very well into the protein workflow. I'll then give you some more details to the system itself and how it works. And then I'll hand over to Susana, who will give some more information as to the study in which we use microfluidic modulation spectroscopy to analyze a set of 13 very similar samples using secondary structure to discriminate between these to determine which of these candidates are viable or not viable for an ELISA screen. So who are RedShiftBio? We're a venture-backed small company based in Boston, Massachusetts in the United States. We're a relatively new company, but we are backed by two of the largest instrument manufacturers in the world, one of whom is Waters, and the other I can't mention. We have raised over $50 million in funding up until this point, and this has allowed us to build the system we will present on today, and to create a global network of sales, applications, and service support. We have a very experienced management team with a track record of success of bringing new disruptive technologies to market, and we have designed the system to be fully automated, not only in the manner in which it runs your samples, but also in the manner in which it analyzes your data. Here we can see an image of the AQS3 Pro. As you may note, the system comes in two halves. The larger unit in the middle is the analytical unit, and at the bottom you can see the autosampler, from which the system takes your samples from 24 or 96 well plates and runs them through the system, through the laser and the flow cell, which are contained at the top half of the larger unit, and the flow cell of which is user interchangeable. And then on the right-hand side, you can see the smaller unit, which contains the inbuilt computer and some other mechanics, such as the pumps. And at the top, there is shown in red is the lid to the degasser unit, in which you can prepare your more viscous samples if you require. The whole system, both units together, occupy quite a small footprint within the laboratory, requiring about 1.5 meters in width and less than 1 meter in depth, depending on the configuration. So what can microfluidic modulation do for your organization? This is a fully automated platform, a benchtop system. So this will reduce the amount of time that the user needs to spend to run these samples. We also, because of the automated nature of the way it makes its measurements, it means that our measurements will be more reproducible. And due to the increased sensitivity of the system in combination, this means we can use microfluidic modulation spectroscopy to detect previously undetectable changes in protein structure. And we can use this information that you can actually acquire in a very wide range of backgrounds in order to guide the product workflow, and therefore reduce the risk of downstream product failures and increase the ability of decision makers to make decisions based on higher quality data. So if we look at the protein workflow, particularly at protein structural characterization, we note that there are very good options in place for all of these, apart from for secondary structure. In this space, there are two main traditional techniques, circular dichroism spectroscopy and Fourier transform infrared spectroscopy. Both of these techniques are very useful in slightly different ways. And in fact, I've used both of these quite extensively myself, particularly circular dichroism. But neither of them give a solution which is particularly an automated solution which solves problems that people experience in the protein workflow. Circular dichroism spectroscopy shines circularly polarized light at a sample of right-handed circularly polarized light and left-handed circularly polarized light, and looks at the differential absorption of the two. And this gives you information as to the secondary structures of your proteins within your samples. But and it's very, very good at giving you information about alpha helical structures and disordered structures, and some information about beta structures, but it's not so good at applying this analysis to the others. It also suffers from a number of other problems, one of which is concentration. We're limited in circular dichroism to about a maximum for a desktop system of about 1 milligram per mil concentration, which is usually quite a long way below what would be needed to look at these samples in situ, particularly in their formulation conditions. And it also suffers from poor compatibility with the background. Being based on absorption of light in the hundreds of nanometers range, this means that we have quite large effects from scattering, particularly from things like excipients in the background, surfactants, and also from aggregates formed by the sample itself. So this means that circular diprism spectroscopy is a very useful technique, but it has quite a lot of constraints on it. Fourier transform infrared spectroscopy is perhaps maybe a slightly better technique in this space because it gives you more information and gives you more signals that correspond to more types of secondary structures. It's also slightly environmentally sensitive in some cases. So the types of structures your secondary structures might be in may give different signals to others. For example, extended amyloid type fibrils or large aggregates will give a slightly different signal to antiparallel beta sheets that may be present within globular proteins. So this can give you that extra level of information. Unfortunately, with Fourier transform infrared spectroscopy, until now, this has been extremely limited, particularly in terms of concentration. So in order to get a reasonable signal for a benchtop system, you need approximately 10 milligrams per mil of your sample, and you need to be very careful with the background subtraction. This has meant that Fourier transform infrared spectroscopy until this point has had quite limited use in the protein workflow and is one of the key problems that microfluidic modulation spectroscopy seeks to solve. These issues have meant that until now, infrared spectroscopy has not been an appropriate technique to apply to the majority of protein-based samples, perhaps at lower concentrations. And so it has not been established as quite as widely a used technique as perhaps it might have been. And these are some of the problems that we seek to solve by bringing this new technique of microfluidic modulation spectroscopy, or MMS, to market. Microfluidic modulation spectroscopy seeks to solve some of the issues with Fourier transform infrared spectroscopy in four main ways. We have a very high-powered quantum cascade laser as part of the system, which has about 100 times the power of a standard light source used in Fourier transform infrared spectroscopy. This gives us about 30 times the sensitivity in signal compared to that technique. We combine this with a high-precision microfluidic flow cell, which means that unlike with Fourier transform infrared spectroscopy, where you have to use two well-matched cells to make your measurements, with this system we actually flow different fluids into the same cell and therefore reduce any errors in that aspect. The system is fully automated, meaning that we run our samples not one by one with cleaning in between, but we load our samples into either 24 or 96 well plates, and we set up our method, and the system will take samples as we wish in the order in which we wish. And then finally, we have quite advanced analytical software built into the system, which means that we can analyze all of your data at the touch of a button. We can actually build it into the methods itself. All of the data is very easily exportable in CSV and in GIF format, and we have an automated and fully customizable report function. As I mentioned, we use a quantum cascade laser as the light source for these experiments. We focus particularly on the amide 1 region for protein analysis, because this is the area of the spectrum which gives us the most information as to the secondary structures of proteins within your sample. We take measurements every 4 wave numbers and interpolate that data to create a spectrum. And due to the optics and the quantum cascade laser that we're using with the system, This extends the dynamic range of microfluidic modulation spectroscopy to much broader than with Fourier transform infrared spectroscopy, which was limited to a minimum of about 10 milligrams per mil sample. With microfluidic modulation spectroscopy, we can measure within a dynamic range from 0.1 milligrams per mil to over 200 milligrams per mil. And furthermore, we have a much higher degree of tolerance to the background. We can measure in very complex backgrounds and in very simple backgrounds and make direct comparisons. And we have no problems with things that would be a problem for circular dichroism spectroscopy, such as different excipients or surfactants in the background, or the presence of aggregates to a limited degree in your sample. To make our measurements, instead of using two matched cells as described for Fourier transform infrared spectroscopy, we instead use just one microfluidic flow cell. This has one measurement chamber, but two inlets, one for sample in its background and one just for the background itself. And I should note that the background does not just need to be your buffer, as is shown on this diagram, but could also contain some other components if you wish to study the secondary structure of your material in the presence of the other. As what we are doing here is we are taking the differential spectrum between whatever solutions are within channel 1 and channel 2. In the bottom left, you can see a diagram of the flow cell, and as you can see, it's in this Y shape. The two inlets for your sample in background and background are at the top, or the narrower parts, and the measurement chamber is at the bottom, with the measurement position shown as a red circle. The flow cell has dimensions of 2 millimeters by 24 microns, with 24 microns being the path length of this cell. And what we do here is we start just by filling the cell just with your background, and then we modulate between the two channels, taking the differential spectrum at each point, multiple times actually, in order to take data points across the amide 1 band every 4 wave numbers, and we interpolate these data to provide a spectrum. This is actually why the technology is called microfluidic modulation spectroscopy, as we are using a microfluidic flow cell and modulating the inputs between these two channels. And this gives several inherent advantages over FTIR. The first of which I've already touched on, being that by using this one microfluidic flow cell, we are effectively getting perfect cell matching, which is a problem with FTIR. Another large problem actually is water subtraction. Water has an absorbance band almost directly in the middle of the amide 1 band, which is the area of most interest for proteins. And this causes a lot of problems with subtraction, and is actually historically why a lot of people have used the comparisons between the amide 1 and the amide 2 bands in the past. We don't have this problem at all. We can control the humidity within the measurement chamber, and the fluids themselves never come into contact with the outside environment, and so we have almost perfect water subtraction here. We also, by using this modulation between the two channels, also account for any short-term drift there might be within the light source, which is again quite a large problem with FTIR, as during the course of what can be quite a long measurement, you can actually have quite significant drift in the light source, and this can cause quite large errors in the spectrum. In order to run our samples, we would simply pipette solutions of sample and then the matched buffer into wells adjacent to one another on either 24 or 96 well plates, in order to run a large number of samples in an automated fashion, either during the daytime whilst you're present within the laboratory, or if you wish, you can load them to run overnight and have all of your data analysis ready for you in the morning. So when we have recorded your data, as I showed you before, taking data points every 4 centimeters to the -1 and interpolating this into a spectrum, we then do some processing on your data automatically as part of each measurement you make. First of all, we take the gradient of that line, or create the second derivative plot of your spectrum in order to discern different features that may be present within your spectrum. We then also turn this upside down and baseline it and compare the spectra across your range of samples mathematically and give you a percentage similarity comparison so that you can very easily use one sample as a reference and compare all of the others to it. And this has a great utility when you are running an automated system, as the AQS3 Pro is, because you'll very, very quickly generate very large data sets, and this allows you to very quickly look at your data and to find out where changes may be happening or where the biggest differences may be between different samples. We then use least squared regression fitting based on models that are from the literature that correlate different signals within the amide-1 band to different secondary structures within your proteins, and very simply output this as a bar chart to provide you with percentage values on the amounts of the different kinds of secondary structures that are present within your sample. This information is output as a series of plots or graphs that can be simply copied and pasted into other programs, and also as a data table at the bottom of your data set. We actually do one more step before we do the second derivative analysis I just described, and that is to transform between your raw differential spectra that you acquire on the system to what we call the absolute absorbance spectra that simply account for the concentration of your sample and something similar to the specific volume that your samples occupy within the solution, something we call the protein displacement factor. And we make these two transformations for every sample you run based on a reference that's either one that we have internally in the system or one that the user generates quite easily for themselves. And we'll give you some extra additional information, including the precise concentration of your sample within an even greater dynamic range than we can do structural analysis for. So here we can do this from 0.01 milligrams per mil all the way to 200 milligrams per mil and above. And we also calculate this factor called the protein displacement factor, which gives you an indication as to the volume that your protein is occupying within its reference or its background. If you then do if you take spectra of a series of samples, perhaps a serial dilution of one sample, it will also output the linearity of the concentrations that you measured for that series as a graph as well. There are several other outputs that are generated by the system, and I should note this is for every sample that you would run or every set of or series of samples that you run, and can be done quite automatically. There are some settings that you would need to fill out to make some of these transformations, but we have a range of example files built into the system, and once you've built your own particular analysis file, in order to run that file, you can either actually include it within your methods, or you can actually run that method or run that analysis just with the click of a button. So some of these I have already described to you. I talked about the second derivative already, which is the gradient of the absolute absorbance spectrum. In addition to this, we actually create another plot, which we call the delta plot, that's shown in the top middle. And this is simply a difference plot between one second derivative spectrum that you might use as a reference, and any others of other samples you have in that series. And this is very useful because it will tell you or show you very clearly where the biggest differences are between your samples. And perhaps will allow you to tell whether or not the differences between two samples are real or significant enough in order to provide you with some information. We can also, because once you have very, very large data sets, it can be quite difficult to look at hundreds of spectra and get some useful information out of that. We can actually just monitor the delta plot at particular wave numbers that the user has full customization over. And we can display this information in what we call the stability plot, which is shown in the top right, in order to allow this data to be more accessible. I've already talked to you about the Gaussian peak fitting, shown in the bottom middle, and its transformation into a bar chart, which very simply outputs the amounts of each secondary structures you have present in your sample in percentage form. And there's also one other very useful output, particularly useful for very large data sets and comparisons within them. And this is called the similarity plot, or the similarity comparison, shown in the bottom left. This is data in which we have taken our second derivative spectra, we've turned them upside down and baselined them, and then we use mathematical analysis to compare these spectra to one another, to just give you a simple percentage value to tell you how similar one spectrum is to another. And again, just to stress, particularly the stability plot in the top right and the similarity comparison in the bottom left are extremely useful outputs when you're looking at very, very large data sets, which are very easily generated on such an automated platform. And so MMS very, very quickly allows you to go from your sample in solution to some usable secondary structure information on your proteins, which have a number of different uses in terms of the protein workflow. These are non-inclusively structure activity determinations. They might allow you to directly measure conformational change that might be related to ligand binding or some protein-protein interactions, or even perhaps some interactions between your protein and the buffer in which it finds itself. These outputs can also be used to give you a good handle on protein stability in terms of formulation development, in manufacturing, and in perhaps batch-to-batch comparisons and comparability. And this information, particularly focusing on transitions between parallel and anti-parallel beta sheet, can give you some structural information as to the nature of aggregation that may be occurring within your sample, and can therefore be used as a measure of stability of your samples. And particularly in this example for this talk, we will be using Microfluidic Modulation Spectroscopy for the identification of biosimilarity and for quality control in batch-to-batch comparisons. So in summary, the AQS3 Pro provides, using Microfluidic Modulation Spectroscopy, provides 5 critical measurements that are related to the protein workflow. Each one of these is already available using one technique or another, but no system that is currently available is able to make all 5 of these measurements on one platform. In addition to this, each one of these systems actually suffers from a number of caveats, be that the limited dynamic range in which they can operate, or perhaps limits in the types of backgrounds in which the samples can be in, or in the fact that they are not automated platforms, whereas here we have a system that covers all of these in one. So we make these 5 measurement types, and this is actually where the name comes from. So the AQS3 comes from the 5 measurement types, which are aggregation, quantitation, stability, similarity, and structure. We measure, or we get an indication as to structural changes that are related to aggregation, by looking predominantly at the changes usually between parallel beta sheet and anti-parallel beta sheet, which indicates the formation of amyloid-type structures. We quantitate the amount of protein within each sample as part of every run, and this is extremely accurate. We actually can do structural analysis across a very wide concentration range for this system, so 0.1 milligrams per mil to 200 milligrams per mil and above. But for quantitation, we have the same upper limit, but we can go down even further by another order of magnitude, down to 0.01 milligrams per mil. We then make three other measurements that are actually all related to structural analysis. So we measured the spectrum across the amide 1 band and deconvolute this to provide us with secondary structure information, as we've just seen. We can then extract some useful information out of this data in order to construct a stability plot. And then we can use this in combination with a range of different experiments, for example, a stress test or something like that, in which you might raise the temperature or change the pH of your sample and watch it fall apart or form different types of secondary structures as part of that process. Or we can use the very useful similarity analysis in order to give us some numerical comparisons between different spectra as well. So we have a range of ways of analyzing this data together. The analysis itself is performed automatically, and as are the measurements from 24 and 96 well plates. And we can equally easily measure your samples in very simple backgrounds and in very complex backgrounds as well. And so with the AQS3 Pro, not only do we have one system that makes 5 measurements that are critical to the protein workflow in an automated fashion from well plates, but we also have a system that makes these measurements across an incredibly wide dynamic concentration range and also an incredibly wide range of different backgrounds as well. And this means that perhaps for the first time, we can use one biophysical technique to compare samples at very low concentration in very simple buffers, perhaps in a phosphate buffer or something similar in the discovery phase, directly to samples at much, much higher concentrations in very much more complicated backgrounds in perhaps the formulation or manufacturing stages. Normally, we do have techniques that do fit to one or another of these stages, but we would normally have to infer data between these different techniques. And of course, as I described a few minutes before, a number of these different techniques have their own caveats, and so this can really complicate these types of comparisons. MMS here will give you a direct comparison using this, the same data and the same outputs between very, very different samples. So thank you very much for your attention so far. I'm going to pass over now to Susanna, who is going to take you through a collaborative project we ran together using Microfluidic Modulation Spectroscopy on the AQS3 Pro. Thank you very much.
Hello, everyone. Let me first introduce myself. I have been working on protein biophysical characterization for over 10 years, detecting structural changes in proteins, mainly by HPLC and other spectroscopy techniques, for both the pharmaceutical and the in vitro diagnostic business. At Biokit, characterization of raw materials employed in our in vitro diagnostic assay kits is essential. Biokit is a working company dedicated to the design, development, and manufacturing of immunoassays. We also offer original equipment manufacturing service to working companies as well as to other manufacturers. Biokit is a privately owned IVD company with more than 45 years dedicated to the research, development, and manufacture of assays and biomaterials for the in vitro diagnostic use. We are located in the province of Barcelona in Spain, where we have newly built state-of-the-art facilities for the development and manufacturing of IVD immunoassay kits. And our products can be found in more than 100 countries, where millions of patient samples are analyzed every year. Now, today I will be presenting a case study of one of our major raw materials, bovine serum albumin, which is better known as BSA. BSA is a globular serum protein of 66.5 kilodalton, which is sourced from animals, and it is widely used for the development of IVD reagents. Typical applications include the use of BSA as a stabilizer or as a blocking agent. And it has been demonstrated that BSA has large variability, vendor to vendor and lot to lot. It is also well known that BSA lot to lot variability is translated into a lack of reproducibility on reagent manufacturing. So different lots of BSA can imply significant differences in lot-to-lot performance of an immunoassay if the BSA is not previously well characterized and selected. Now, for our case study, for a specific reagent, several lots of BSA from different vendors were purchased and characterized in order to know if we could predict its performance. Therefore, analytical characterization of this raw material started in order to understand if there were any structural changes or traits able to explain why some BSA samples worked and some did not. So samples having a good performance in the IVD assay were labeled as okay, while samples not having a good performance were labeled as not okay. There was one sample which performance was in a borderline and crosses were assigned to it. So sample performance labeled with a minus mean that they have not been tested in our assay yet. As the tested HPLC analytical techniques did not clearly explain BSA performance, then we started a collaboration with RedShiftBio in order to try the innovative AQS3 system, which mode of action and benefits has previously been explained by Dr. King. This system is powered by Microfluidic Modulation Spectroscopy. It is an automated, easy-to-operate system which has a powerful laser and high-precision microfluidic cell for superior sensitivity analysis. Actually, a recent publication compared the sensitivity of the microfluidic modulation spectroscopy to conventional Fourier transform infrared method. And the outcomes show that microfluidic modulation spectroscopy secondary structural analysis was comparable to that of FTIR. They also demonstrated that the repeatability of this novel microfluidic modulation spectroscopy technology was superior to the Fourier transform infrared method, and the sensitivity was 30-fold higher, and that was especially true for the lower range of protein concentration. Now, coming back to our study. So one of the BSA sample, which has proven to perform okay in our assay, was selected as reference and scanned at different concentrations in order to study the concentration effect on sample structure. Then, the obtained equation was employed to determine BSA concentration for the rest of the samples. Now, note that the relationship between BSA concentration versus signal was larger than 0.999, which demonstrates the reproducibility of the AQS3 system. Well, the given concentration of all BSA samples was 10 milligram ml. However, after employing the previously generated equation, the concentrations were calculated. We also obtained the displacement factor, which were approximately the same for all samples. And also, we obtained the similarities, the sample similarities. It means how similar the study BSA samples were with respect to the selected reference BSA. And if we look at this table, we can notice that the samples performing okay, they showed to be more similar to the reference sample, they are highlighted in blue, than the rest of the sample. And this similarity showed to be larger than 98%. As when performing microfluidic modulation spectroscopy, sample background is constantly being subtracted, then absolute infrared absorbance scans are gathered. For our study, 3 replicates per sample were performed, and at first glance, no differences are detected. However, if we zoom in, 2 differentiated groups can be observed. As mentioned before, 3 replicates per sample were performed, so they were averaged, and the internal replicate reproducibility was measured, and that demonstrated to be larger than 99.5%. After averaging each sample spectra, the convolution of the infrared spectra was performed, and the 2-dimensional structure contribution plot comparing relative amounts of alpha helix, beta sheets, unordered and turn structures were obtained and compared to the sample used as reference. Well, the largest observed difference between samples was smaller than 2%, which is very difficult to discriminate when using traditional technologies, but it's possible due to the extremely high sensitivity and reproducibility of microfluidic modulation spectroscopy technology. As we can see here, one particular structure contribution demonstrated an interesting trend with respect to sample performance, the unordered structure. And unordered structure has been related to protein denaturation. For our case, this structure contribution seems to explain performance for our IVD assay. As this study showed, it is clear that structural differences exist among these BSA samples, and delta of second derivatives accentuate these structural differences, which in our case show to be very small, like I said before, smaller than 2%. And it is clear that some BSA are more similar than others. Similarity plots show that the OK BSA sample difference were smaller than the observed for the rest of the samples. That is why understanding and managing structural differences is essential for product quality. And as a wrap-up, during this study, using microfluidic modulation spectroscopy, we were able to detect smaller than 2% structural differences for different BSA samples, thanks to the high sensitivity of the AQS3 system. And with this, I will hand the microphone to Dr. King.
Oh, thank you very much, Susanna. And thank you for your clear explanation of the data. That was really fantastic. I hope you can see that that here, just using this very quick study we ran, we were able to actually acquire all of this data from 2 24-well plate preparations. You could actually easily collect all of this data and replicates of each set using 1 96-well plate, and it could run during the course of 1 day, and all of this data could be analyzed and the outputs we have shown in this talk provided to you as part of that run. So, and additionally, by using the outputs that Susanna has just been taking us through, I hope you can also see that rather than having to trawl through a lot of different spectra and to understand where the differences are between them, MMS, using the AQS3 Delta analytical program that's built into the system, really allows you to access this data much more quickly in a range of different ways. And here we could see that we can actually not only get this information out very quickly, but also confirm it using each one of these different methods. So we could compare it numerically here with the similarity analysis, just giving us a percentage score comparing the reference to each one of those samples. We could also look at the secondary structure outputs themselves in bar chart to very clearly see, particularly with the disordered spectra, that those which show a smaller amount of the disordered structure were clearly the candidates most similar to the reference. And we can also use the delta plot comparisons that were output in the stability plot that you just saw on that last slide, which very, very clearly shows that there are two groupings of samples, not just when you look at just one type of secondary structure, but here, because we're using the full spectrum, across all the different types of secondary structures present within those samples. So I hope this study was a really good illustration to all of you as to how the AQS3 Pro using microfluidic modulation spectroscopy can be used to accelerate the development of your research by using the automated workflows from 24 and 96 well plates. We reduce the user time required to run these samples quite considerably. In order to run all of the samples shown in this study, you would probably be looking at sort of 5 or 10 hours of active user time using traditional techniques that are based on cuvette-based systems. Here, because we're running these things from 96 or 24 well plates, which we can load very easily. Also, which I did not mention earlier, just by following a diagram that the software shows you once you've built your method. You can simply press go, include the analysis in your method if you wish, and allow the system to acquire your data and process it to give you these very clear outputs in a much, much shorter time period in terms of user time at the system. So, in addition to this, as I've covered, the MMS will also make these measurements across a much wider dynamic concentration range than either of the two traditional technologies in this space, which are circular dichroism spectroscopy and Fourier transform infrared spectroscopy. We not only the concentration range of both of these techniques, but we also can measure in a range of backgrounds that are particularly inaccessible to circular dichroism. We can measure in backgrounds that contain, for example, organic solvents, or they might contain difficult excipients, polymers. They might contain something like surfactants, or even aggregates of proteins themselves. All of these things can cause problems when you're doing spectroscopy because they will scatter light. But with infrared spectroscopy, particularly because we're using such high wavelengths, we're using wavelengths in the micron range here, and also uniquely due to the optic setup of the microfluidic modulation spectroscopy platform in combination with the very powerful quantum cascade laser and the microfluidic flow cell, this means that we do not have these problems and can measure in an extremely wide variety of different backgrounds. And as I said, this means that not only do we make 5 critical measurements for the protein workflow, but it also means that these measurements are across an extremely wide range of samples, allowing us to direct between samples, perhaps at the discovery phase and those in the formulation or manufacturing phase. And all of these things will, we think, allow a microfluidic modulation spectroscopy to become an essential part of your toolkit, your biophysical toolkit, and with the aim of course, de-risking your drug development processes and improving the decision-making process based on higher quality and more reproducible data than was previously available. So thank you very much, everyone, for your attention. And thank you very, very much, Susanna, for your excellent presentation and for allowing us to show some of your data to a wider audience like this. Thank you very much. I just wanted to mention that if you would like some more information about RedShiftBio, you would perhaps like to read some of our application notes or white papers, or some of our more recent publications. We have several really fantastic publications actually from the last 2 or 3 years. All of these are available at our website at www.redshiftbio.com. and we please invite you to visit us and ask us any other questions you might have. And please, if you have any questions now for either of us, please do ask them. Thank you very much.
Thank you so much, Susanna and Patrick, for that fascinating presentation. And now we're going to open it up for some questions. Here's the first question. For aggregated samples, can we get any information about the aggregates?
Thank you, Melissa. Yes, very good question. Yes, we can. So we can actually measure several parameters that can give us some information about aggregation processes that might be occurring. You can use the quantitation part of the measurement to first of all know whether or not the concentration of your samples is decreasing between replicates. We sample in the wells from the bottom, so if we see some variation between subsequent replicates, that can tell us that some aggregation has occurred or is occurring during the time course of the measurement. We have a flow cell that has a path length of 24 microns in line between the well plate and the measurement chamber. We actually have a 12 micron filter, but as long as the aggregates are smaller than this, there is no reason that they won't pass into the chamber. And because we're using quite wavelengths of light in the micron range, we can get some information on the actual structures of these aggregates themselves.
Oh, excellent. Okay. All right, here is another question. What are the main application areas for MMS in the protein workflow?
Oh, yeah, also a very good question. Well, as one of the slides I presented might have shown you, we think microfluidic modulation spectroscopy, with its very broad concentration range and its ability to measure in a very wide range of backgrounds, is a very suitable technique for a number of different applications, particularly in that we can compare directly between samples in very simple backgrounds and perhaps at low concentration with those at much higher concentrations and much more complex backgrounds. But there are many applications that we can apply this technology to. We are simply looking at structural change. This may be perhaps due to some stress conditions that are applied to a sample to assess its stability. You might want to study the structure of your protein samples in different formulation conditions, so you could use this in formulation studies. You could use the similarity analysis to give you numerical comparisons between batches, perhaps, in manufacturing. And as I've discussed, it can be used in order to look at your sample's aggregation profile or process. We could use it, again, with the similarity analysis to look at biosimilars or to look at batch-to-batch reproducibility, as we did in this particular example. And there are a really wide range of other applications as well.
Okay, excellent. Another one, this one's for you, Patrick, again. Is there any size limitation of the protein? What is the smallest protein size that you've tried?
Oh, okay, yes, a good question. So we are looking at infrared absorbance on the peptide or protein backbone. So we're looking at the carbonyl stretching frequencies, which change depending on the types of structures, secondary structures that your protein is forming or your peptide. There is no distinction between the two. They both have the same backbone chemical structure and therefore give very, very similar signals. And there is no actual size limitation on the size of the protein itself, either at the high end or the low end. All we really need is enough of the peptide or protein backbone in the sample to give us the absorbance that we need. And that's what our concentration range is based on.
All right.
And perhaps I could just add.
Sure.
Sorry.
Absolutely. Sorry, Patrick. That's okay.
Excuse me. Perhaps just to add, our specifications are based on a lysozyme protein. Give you a reference.
Okay, thank you. This question is for Susanna. Since BSA can bind fatty acids, is a variation in BSA's secondary structure observed plus or minus fatty acids?
Hello. Yeah, that is
Susanna.
correct. Yeah, hi. That is correct. And we have observed that before. However, for this particular experiment, we did not, let's say, study the fatty acids, so we could not correlate it to the structure right now. But yeah, that is correct. BSA can do that.
Okay. Thank you very much. I believe this one is for Patrick. What type of variance do you see with run repeats versus run replicates?
Good question again. One of the major advantages of MMS being an automated platform and in terms of its optics is the reproducibility of the system is incredibly high. So between replicates of the same sample, say prepared in the same well, the reproducibility will depend on your concentration, but we would expect approximately 98% at about 2 milligrams per mil. And I have seen incredibly high reproducibility at higher concentrations. For example, I've seen about 99.9% for a MAB sample at about 100 milligrams per mil. In terms of run repeats, that would depend on what is actually occurring with your sample. And it's actually a very good experiment to look at. So in order to actually know whether or not we are actually measuring changes between samples, what I would like to do is to run several replicates of one sample, establish the internal variance of that measurement, and then compare that to another sample having done a similar number of replicates. We can output that on the delta plot that I described to you earlier. And this will very, very quickly tell us what the bounds of the internal variance are for one sample and plot any deviations that occur outside that range for another sample. So not only will that tell you that your two samples are statistically different from one another, but it will tell you in what regions of the spectrum these differences occur. And then perhaps one final thing to note here is also that the AQS3 Pro with your samples from well plates, but the actual robot that does this is actually fully addressable, and we can run our samples in any order we wish. And that means that we can actually do analysis of samples with perhaps pause steps, which you can include in the methods, or in any other type of permutation you might want. And you can set up an automated run where it will sample from one set of wells, and it might analyze the rest of the plate and then come back to the first set of wells and take another replicate for you. So if you wanted to study a process that might be happening or changing over time, you can do that sort of experiment.
Okay, thank you, Patrick. I think we have about time for maybe one or two more questions. How many samples could be run in a single day on the AQS3 Pro?
Okay, so we using the 96 well plate, we could probably run about 30 samples in one plate preparation and one method setup, and that would complete in probably less than 24 hours, about 30 samples.
Wow, okay, that's great. All right, this is our last question. What are the greatest differences between circular dichroism and infrared analysis for proteins?
Oh, right, okay. So circular dichroism and infrared analysis are two central techniques for secondary structure analysis for proteins. They're both fantastic techniques. I've used circular dichroism quite a lot myself, and it's very, very useful. It gives you really, really useful information on your samples. The way it works is it shines circularly polarized light at a sample, both right-handed and left-handed, and it looks at the differential absorption in across the range. So very much like MMS works in one way. But the problem with the CD is that the type of light that we shine at the sample is in the UV range. So the light has a wavelength on the order of a few hundreds of nanometers. And this is a problem because objects that might be in either your background, something like surfactants or excipients, or even aggregates that are formed by the proteins themselves, can actually scatter some of that light, and they do so more towards the left-hand side or the lower wavelengths of your spectrum, and this can cause some artifacts and some deviations in that spectrum that you may account to. Well, it may give you incorrect information as to the secondary structures. Infrared analysis uses much larger wavelengths, so much longer wavelengths, sorry. So we're looking at wavelengths in the micron range. And so scattering from the objects I mentioned are much reduced. And so this is the main reason.
Okay, all right. That's about all the time that we have. I'd like to thank again, a big thank you to Ms. Susana de Jesus Acosta and Dr. Patrick King for this fascinating presentation. And thank you to all the participants for being a great audience. Be sure to check CNEN or CNEN online for information on the next edition of CNEN webinars. Thank you On24 for technology and production services, and a special thank you to RedShiftBio for their sponsorship that made this interactive webcast possible. For CNEN webinars, I'm Melissa O'Mara. Goodbye.